So, welcome all. Yeah, thank you so much, Sandeep, for joining. Yeah.
You're welcome, Prash. Hi, how are you?
You're all well, Sandeep. So, Sandeep, please allow me to introduce, Manjari, who is our Secretary of Bioclues. We also have another Sandeep, who is our, Vice President for Entrepreneurship Chair, who will be introducing you. Okay. Yeah. And
Thank
the
you.
program is, we'll have your talk for 20-25 minutes and 15 minutes for Q&A. Thank you so much.
Thank you very much.
Sandeep, we can get started at 7:02. Okay, let others join. There are a few others expected. There are, some of them are
Okay.
also trying to attend through YouTube as well.
So, welcome to Bioclues, Mr. Sandeep. So today
Thank
we are
you.
here to listen and learn on a NextFlow, which is a NGS-related technique, which is very important in the present era. Today, most of the studies in human genetics are heavily dependent on the NGS analysis. And because the most of the genetic diseases are caused because of the multiple genes which are involved, so normal traditional approaches to study the genetic diseases is not so valid. So today, whatever we have, the NGS is a workhorse for most of the human genetic studies. So our speaker, Mr. Sandeep, is working as a senior grade lecturer in the Department of Bioinformatics, Manipal Academy of Higher Education. So Mr. Sandeep's research areas include genomics and proteomics, development and automation of pipelines for DNA-seq and RNA-seq, big data automation and visualization. So Mr. Sandeep, stage is now yours.
Thank you very much, Sandeep. I hope everyone is able to hear me clearly. Thanks for the very kind introduction. Let me share my screen. I have shared my screen. Is this visible?
Yes, please.
Yes.
Perfect. Thank you very much. Thanks again. At the outset, before I even start, I would like to thank Prash. He's been very kind to ask me to speak to all of you. I think Bioclues is a great platform where people, like-minded people, can actually interact with each other and learn from each other. Thank you again. Thanks, Prash. Thanks very much. Something about myself. I am, like Sandeep really nicely introduced me to all of you. I work in the Department of Bioinformatics as a faculty here, and my primary research area is genomics. I have extensively worked with micro analysis of microarray data, and gradually I've moved on to analyzing next generation sequencing data. So I'm assuming most of you are bioinformaticians here, and all of you are very familiar with an image which looks like this. So most of our analysis involves these complex workflows, which is nothing but involving a series of tasks. Like, for example, this is a workflow for analysis of ancient genome, where we start with collecting the data, which is then cleaning it up, like, for example, QC, and processing the data. The entire process here would be called as a workflow or a pipeline. And as all of us know, these workflows, they require multiple software packages, and sometimes we need to run them on different computing environment. Could be a desktop, maybe sometimes we need to run it on the cloud or any other computational setup that we have. So as of right now, I extensively use these pipelines. I mean, we run it for our regular analysis, and how we have used it till now. I think I have a hand raised. Okay, I think we'll take the doubts a little later. So as of right now, what we do, these are used in, they are joined using scripting such as Bash, or could be in Python as well. Now, where does NextFlow come into play? And the reason we're talking about NextFlow. Now, while we connect the system, there are a lot of issues with the system, like, for example, dependency errors. And what helps us in such a scenario is the concept of a workflow management system. So there are plenty of workflow management systems such as SnakeMake. We have SnakeMake. We have Galaxy. Even Galaxy is a GUI-based workflow management system. And very recently, we have NextFlow. Now, NextFlow is specifically tailored to manage computational data analysis workflow. And this involves, NextFlow is capable of doing runtime management, it's capable of software management, and many other aspects. There is some example, I think just go one slide up. So basically, in the area of bioinformatics, we see that the same application deployed in different environments produces different results. And this is very profound. Actually, this is a published study. If you go back, there's a study which was published in Nature Biotechnology, where actually people, the NextFlow group itself has published the study, and the entire tenet of NextFlow being developed is based on the study, where when they saw, for example, annotation, the annotation of genomes using the companion pipeline, it has given a different set of results in a different computational setup. Expression of genes have given a differential expression with Callisto, also has given a different set of result. But when the analysis was performed using NextFlow as a system, we see that in different environments, also computational environment, the results are consistent. And this was extremely powerful. And these are the challenges that NextFlow aims to solve over time. Basically, reproducibility, like I just demonstrated, reproducibility is a huge major challenge, and replicating results over time is a headache. We could spend more than 250, 300 hours just to reproduce some data which was analyzed in the lab. And of course, the script that we can run on a laptop we may not be able to execute the same script on HPC or on a server with the different OS. That's a major issue that we have. And also, some of the tasks, if you run on HPC, it can spawn from approximately 100 to thousands of jobs over different clusters. And very importantly, most of the tasks that we perform as bioinformaticians, it's a mashup of different tools. Sometimes I may have borrowed a small script from a friend of mine, or I could have downloaded a block of code from Git and then try to build this workflow. So to overcome these challenges, specifically, we can use the system NextFlow. This is a beautiful chart on NextFlow. So NextFlow is actually a scalable and reproduce. It can produce scalable and reproducible scientific workflow. It essentially it integrates various software packages and also environment management systems such as Docker, Singularity, Conda. Most importantly, this is used for all existing codes and existing scripts, existing pipelines that we have in different scripting languages. It could be in Bash, it could be in R, it could be in Python, irrespective which scripting language, we can seamlessly couple them and execute them. And once we have this NextFlow script set up, we could run them on any major platform. Could be AWS, could be Azure, on Slurm, Grid Engine, Google Genomics, could be on any set of cloud. And more importantly, NextFlow is also backed by a very strong user community. And I will be discussing in the next few slides the package specifically for bioinformatics use called NF Core. Some of the features that we can get from NextFlow would be fast prototyping. So if we have existing set of scripts, we can quickly scale it up for analysis. Reproducibility, like I mentioned, using containers such as Docker and Singularity, as well as package manager Conda. It is portable. We just discussed we could run it locally, as well migrate the same thing on a HPC cluster as well on the cloud. More importantly, the entire workflow has the intermediate results produced, and these are automatically tracked, which enables a continuous checkpoint system. So at any point of time, for whatever reasons, it could be an accidental stroke of a key, could be a power failure, could be a network issue. It allows us to resume the execution from precisely the point it had stopped for any given reason. I'm not going to spend much time on this aspect, but the whole NextFlow is built on this premise of processes and channels. Our processes are nothing but these individual tasks where the entire pipeline is made by joining together different processes. And each of these processes can be written any scripting language, could be like I said, Bash, Ruby, Perl, etc. Now the processes are independent. For example, like the first step, fast QC. For a set of fast Q files, I have to run fast QC. Now this is the process. So what does that do? It takes my fast Q file and it gives me the QC values. So that's my task one. And similarly, it does for second fast queue, third fast queue. And probably if you have BAM file, we want to index the BAM file. So that is the process. Now, what is a channel? Now, channel is the terminology that NextFlow uses to connect different processes. It's basically it's basically how NextFlow handles the data management, allowing the complex the tasks in hand to be split up and then run in parallel. So the output of a task A could be input of task B, and that is how channels manage the data. So in the first in, first out order. The focus, it's a very short time that I have, 25 minutes. I don't think so I'll be able to do so much justice to NextFlow, and I'm not an expert at that as well. But personally speaking, I have used NF Core, and this is what I'm trying to do currently in our institute as well. Like Sandeep mentioned, I do work with development of pipelines, but till very recently, I never used a pipeline management suite. Just started using NF Core to be very specific. I'm not an expert at NextFlow, and NextFlow is built on Groovy, and I don't know Groovy. So, but without knowing Groovy on which it's built up, I am still able to use NF Core to execute very high quality, build and execute very high quality pipeline. Now, what is NF Core? I think this is very important to all the bioinformaticians and as well as anybody working in the area of life sciences. So this is a community-led project to develop a set of best practices, best practice pipeline using NextFlow. If we go to the NF Core platform, we will find highly optimized pipelines with high reproducibility and thoroughly tested pipelines. So these are all the NF Core pipelines, like I mentioned, are portable. They're very well documented, and there's a easy to follow workflow. So this is a screenshot of the NF Core platform. So if you go to the available pipelines, as you can see, let me document here. So if you go to the NF core, you'll find these platforms. For example, AmpliSeq. There is this is the eager. That's the one I showed you for ancient DNA. There is epitope prediction, viral recon, RNA fusion, and this is a screenshot. So I've missed quite a lot of pipelines here. But I'll just visit. We'll visit the site once the presentation starts. So the task here would be like, for example, if I want to use the RNA-seq pipeline, I just go to NF core, look for RNA-seq, and then follow the instructions there. Essentially, before we even do that, we need to install NextFlow on our system. It's as simple as this command here, curl -s. Basically, get the NextFlow on the machine, and once you have NextFlow on the machine, you can set the path and then do a pip install NF core. Once the NF core is installed, I think we're good to go, and we can launch any pipeline in the NF core by using the specific command NextFlow space run NF core and name of the pipeline. So a very simple example of NF core. If we enter this command NF core space list, we'll get a list of the pipelines available on the terminal itself, and whether we also know whether it is locally installed or whether it is not. So the beauty about any NF core pipeline is we need not have it locally installed. We could directly run it from the cloud. We do not have to download any databases. For example, GRCS 37, 38, the annotation files. Everything is the links are pre-embedded in the pipeline itself. It'll fetch on the fly. We have an option to save it on local system or run it on the fly, in the sense delete it after the analysis is done. So once we identify what pipeline we want to use, we could proceed either by running it directly or running it locally. For example, to run the pipeline, this is an example of running this bacterial map pipeline. So we could just NextFlow run space NF core / backmap, give the version and help. It gives exact instructions how to run the pipeline. A beautiful training material, Sequera Labs. If you could just visit this website, training.sequera.io, you'll find the entire training modules for NextFlow. This is a company that actually started building NextFlow. They're not new. They're approximately, they've been there for the last 15 years. But since NF4 came into play, a lot of bioinformaticians started using it, and there's traction on Sequera Labs. Something which I can't do justice in this 20 minutes, 25 minutes that I've got. While preparing for this presentation, I happened to visit NF core website, and I see that there is a training for NF core scheduled in the month of October. I do believe this is free, and there are different time zones. So I would suggest anyone, probably everyone who's here today, could make use of this opportunity. If I'm not wrong, it's around 2.5 hours every day for 3 days. You could please check the schedule. It's given there, I think, for 2 days, and make use of this opportunity. I have shared some resources here. I'll share the PowerPoint with all of you. I'll give it to Sandeep or Trash, and they will distribute to all of you. So with this, I like to thank everyone. And before I even conclude, I want to demonstrate something on the computer. So we use up the 25 minutes, and then we move to question and answer. Let me new share just a second. Yes. So right now I am at NF core, and if I go to NF core, you see this view pipeline. This is where we need to go. And if in available pipeline, we see a list of different pipelines. For example, if I want to do anything with RNA-seq, I could just type RNA-seq and it gives me a list of pipelines involving RNA. For example, here RNA-var is a RNA variant calling pipeline, and this was published 3 months ago, and this is still version 1. And for example, this is a single cell RNA-seq. Now, this is what I'm currently trying to implement on my end. And this, again, this is version 2, and this is also implemented 3 months ago. So one of the most popular pipelines that they have for RNA-seq is actually RNA-seq. So this is an RNA-seq analysis pipeline using STAR, RSEM, HiSat2, Salmon, with gene counts and extensive quality control. For example purpose, let me take you through NF core RNA-seq. So NF core RNA-seq, it gives us the introduction to RNA-seq. There is also the online links to tutorial materials, and it tells you what exactly the RNA-seq does. Here there's a list of tools and the entire setup, that how the pipeline works, starting from fast QC to adapter trimming, removal of contaminants, removal of ribosomal RNA, and then we have a choice of alignment. Of course, we have STAR, then HiSat2 as well. Regular processing like sorting, indexing, and finally it also does differential expression and quantification also. So once we identify this pipeline, we can use the usage docs and exactly run the pipeline. For every single pipeline, there is a test data set. Once we log in here, there will be a test command. We run the test command and we identify whether the pipeline is set up or not. Follow the sheet. There are data files also that is provided, and we can easily determine if we have successfully got the pipeline configured or not. And most importantly, before we end, this is the output. What kind of output will our RNA-seq pipeline give us? And if you scroll down, this is actually an output of their sample data. So right from FASTQ to the intermediate files, the alignment and quantification files, and again, using HiSat, all the data that is, all the statistics that come out of the sequencer, and intermediate steps, and finally, including visualization of visualization using different tools. So let me show you. Yeah, these are the the intermediate plots generated during differential expression analysis. So I think this is it from my end. I have I hope I've done justice to the time that is given to me, and I really thank Prash again. And most importantly, I thank Bioclues as an organization for giving me this opportunity to speak to you all. Please feel free to connect to me. My email address is sandeep.mallya@manipal.edu, and I'll definitely respond back. Thank you very much.
Wonderful, wonderful talk, Sandeep. Thank you so much for your time. Sandeep, please, take the Q&A, please. Yeah. Sandeep Kumar.
Sandeep, yes, I can. Yeah, I am looking at it.
Yeah, so we'll help you, Sandeep. I'm sorry. So Jay Kishan asked if I run one pipeline in his data backed by cloud instance, could machine computational power be utilized, or how is it? Yeah.
Yeah, hi Jai Kishan. It depends on, for example, if it's a simple pipeline, like for example, the RNA-seq test pipeline, I could run it on my laptop. It uses my resources. But at the same time, if I deploy it on the cloud, it will not use my local resource. Of course, it will be using the cloud resource. So the answer is it will use the computation power of your cloud, not your local instance.
Yeah,
So I
wonderful. So
hope that
please feel
answers.
free to ask questions. Yeah. Uh,
Yeah, I think Bilal already answered the question. I guess it depends where you're running the pipeline. Absolutely correct, Bilal. It definitely depends on where you're running the pipeline. Like I mentioned earlier, there's a beautiful training program. It's free, and I think it's very convenient time. It's morning 8:30 Indian time. If you go to the Asian time zone, or we also have the European time sometime in the evening. I didn't check, but the American time is at 10 o'clock in the evening. So I think we have a beautiful time slots here, either early morning or late in the night. So whichever works for us. In this community, I think it will be extremely useful if many of us get trained in this, because we could easily share our codes. Any custom pipeline can easily be shared among each other. I think it's a fantastic opportunity for all of us,
yeah.
for a free training.
Sandeep, I have a question. Sandeep, does it also run parallel? I mean, could we also run MPI run in enough code?
Yes,
Yeah, okay. Yeah.
absolutely, yes. So with the same set of the best part is, like, I personally I have no access to a multi-node system, or neither I have the requirement. But while going through it, it clearly says that even though I write the configuration on my laptop, it just when I execute it, so I go NF core space NextFlow space NF core slash the pipeline name, and just give the argument saying on how many nodes you want to run and execute it. It automatically deploys on the multiple nodes as well. That is correct. It does that.
Wonderful, wonderful. Yeah. Any questions, please? Anyone else? Yeah.
There's a question, is there any tool in the NextFlow that provide option to connect with the cloud instance? Um, yeah, I think I'll add a link to one of the tutorials. Okay, a brief history. I think I still have time, so its schedule is till 7:30. Uh, a good friend of mine called Satish. He's currently in the US. He's a trainer with CDC. He's the one who introduced me to NF Core. I'm not an expert at Nextflow. Like I am not in a position to write my own pipelines, but I do deploy the existing pipelines on NF Core. And he has a training module. I've taken a lot of materials from him. It's of course open to share. So I'll share that as well here. Um, Over there, he in the training module, there's a service called Gitpod. I'm not sure how many of us are actually aware of Gitpod. Gitpod is a free cloud instance for training purpose. You can instantly fire up Ubuntu system and work on it. So it gives as a student some 50 hours per month free cloud resource. So anything for training or practice purpose, that's one of the best and easiest way to fire up the terminal. I'll just share the link. The reason I'm saying Gitpod is because um, it's very simple. It's a total 9-hour series because it's a structured workshop. It's a 9-hour series with introduction to NextFlow and the entire you know the the, the workshop is running on Gitpod, which is a cloud-based system. So probably Jai Kishan, that will answer your question. If you could just follow that workshop, it will tell you how to connect with the cloud instance. I'll share the link. Prash, I'm not sure how do we share. I could share the presentation. Should I send it to you, and will you be sending it to all
Yeah,
the members?
yeah, you can. You're in the South chapter group, right? So you can convert into PDF and share it in South chapter.
Okay, I'll do that.
Yeah, yeah. And then I'll forward it to North chapter and International chapter. There are three other groups as well. So yeah.
Okay, great. Anybody, any more questions?
Yeah, please feel free to ask.
Hello.
Yeah.
Hello.
Yes. Hi.
Hello. Hello. Hi, sir. Nice talk, sir. It's Bilal here.
Hi, Bilal.
Sir, I have one question. Like as you mentioned, and like one of the main feature of NextFlow is like we can reproduce the same result. Like several researchers who have been performing the result from different pipelines, they will be getting different results. But via using NextFlow, we will be having the same like result at the end. So is there a chance we can like have a paper which will be having all the like pipeline mentioned as a protocol, and we can replicate on our own system rather than using AWS like Slum? That's some kind of work like without using AWS or Slum. Can we like a small chip data set or like some kind of small RNN data set where we can just want to replicate and it is published somewhere so we can exactly see the protocol?
Uh, Sorry, Bilal. It was your voice was breaking. What I understand is you want to know if there's a small data set that you could replicate on
Yes,
your
sir. Yes,
system.
sir.
Uh,
Yes, sir. Yes.
do you mean to say a small data set which is published and not a part of NextFlow, or you anything existing NextFlow?
Sir, what I meant is a small data set which has been published in some protocol, like the people who have already published
Mhm.
it's at some manual. So we can just replicate it by using our small limited resources on our computer.
That I am not sure, but definitely, like I got the statistics. If you remember seeing one graph, that's from a paper where they have taken the existing data set and they've analyzed. And why? Okay, what happens? See, the common challenge in bioinformatics we find is the dependency. So
Right.
so any workflow that we use, it is a series of multiple tools. And the tool, like for example, if I use HiSat 2, so I may be using HiSat 2 version 1, and you may be using HiSat version zero point 0.8. Now your results are a combination of HiSat 2 version zero point 0.8, SAM tools version zero point 0.6. But when I am deploying it by default, when I pull or when I install using Conda, by default it gets the latest version gets installed. So even though I've used the exact same tools, exact same flow as you have done, the results will be different. See, the core result will not change. It's just that among if you have reported around 300 genes that were significantly differentially expressed, I may also get 300. But the core set of 250 will remain intact, and you will have 50 a unique set, and I'll have 50 unique set. If the core set changes, then definitely there's a problem with the analysis. Definitely there's a problem. We cannot have the core set changing. But if the core set remains intact, I think that is very acceptable. So instead of trying
Uh,
to focus on replicating the published result, I think our focus should be more on implementation and trying to get a pipeline executed so we can use it for any purpose in the future.
Sure, sir. So last thing, as like you showed in the PPT also, like there are different versions of pipeline. Like I have tried implementing one. There are different versions of pipeline.
Yes.
So how come it is like it might be the same thing again because like there are different versions of pipeline and people are executing different pipeline for their analysis. So
Yeah.
there might be still chances of this error, right? Like should we
Yes.
have to mention this in like while writing
Correct.
paper or something?
Yeah. So while writing paper, you can mention the version, because while you install or while you deploy a particular pipeline, for example, NF core slash RNA-seq, you give a version number also along with -v 2.0.1. So if I have executed my analysis using 2.0.1, you can always reproduce using 2.0.1. because you have an option to exit, and that version of the pipeline will have the same set of tools, so hence making it reproducible.
Okay. Thank you,
Because
sir. Thank you so much.
yeah, after 6 months when I go, it will be version 3.5.1. So if you had noticed it, good observation. So if you had noticed it, certain pipelines which are not very frequently used by the community, they're still in version 1. Whereas commonly used pipelines such as RNC kits in version 3.5.7 or something like that. So
Right.
that gets constantly updated compared to some other pipelines which are not frequently used.
Sure, sir. Thank you, sir. Thank you for your time.
Welcome. So there is a question, can I use any NF core pipeline and publish the pipeline on your own data? Yes, of course. If you see the NF core documentation, they have given, there's a paper published for NF core, I think it was in 2019 if I'm not wrong. So you just cite them and yes, you can use. So the link which I share, it has the citation also. You cite the NF core paper. Can I modify code? Absolutely yes. You can modify the code of NF core pipeline, and all that's where you need a little bit of skills in NextFlow. And if you just need to change the configuration, there's a configuration file. You can easily modify the configuration file. That does not need much knowledge on the programming aspect using NextFlow. You could easily do that. So we have a message from Bioclues, all of us to switch on the camera for a quick photo because I think we're going off in the next 4 minutes. I have my
So we
camera
don't
on.
want to, yeah. So Sandeep, we don't want to lose this opportunity to have a smiling photograph with us, Sandeep Mallya. So yeah. So thank you so much, everyone. Yeah, Selva, Anuj, Jaraman sir, Pranavati Ani of Pondicherry, Manjari, our secretary, Subhu, our advisor, Shravya, Soumya, please, yeah. Akshay, yeah, okay, yeah. So, yes, as always, I'm a very good photographer. Okay, yeah, thank you.
Thank you very much.
1. Yeah, thank you. Thank you so much, Sandeep. So friends, I think, we should give a big round of applause to Sandeep Mallya and as well as Sandeep Kumar for hosting this. Thank you so much. Yeah. So thank you. See you next time.
Thank you very much. Again, thanks very much. I think we should do this more often. Thanks very much. Bye-bye.
Thank you, Sandeep. Thank you.